Bangalore chapter. It gives me immense pleasure to welcome you all for this webinar on Know Your Microbiome. As healthcare professionals, dietitians, and advocates of better health, it is essential for us to stay ahead at the forefront of emerging sciences. The human microbiome is one such groundbreaking area that has reshaped how we approach nutrition, disease management, and overall well being. Moreover, we are witnessing microbiome to be the rapidly evolving field of study and how it is impacting the health and wellness. And to take a deep dive into this topic, we will, we will have to, enhance our knowledge by attending sessions and by reading it more about it. And today, on behalf of Indian Dietetic Association, we have actually, curated a session which is going to enhance our knowledge on this field. And we are honored to have Dr. Debojyoti Dhar, co-founder and director of Leucine Rich Bio with us today, who will provide us profound insights into the role of the microbiome in personalized nutrition and its potential to improve health and outcomes. Finally, we will first start with this session and then we will wrap up with the emerging Q&A session and the vote of thanks by Ms. Shalini Arvind, co-convener and our chapter co-convener of our chapter and chief clinical dietitian from Fortis Hospitals. I would like to thank all of you for taking time to join us today. Your participation reflects your dedication to advancing the field of dietetics and bringing the best to your patients. So let us take this opportunity to learn, interact, and take back valuable insights. Without further ado, let me introduce Dr. Debojyoti Dhar. Can I have his intro slide, please? Dr. Debojyoti Dhar is a co-founder and director of Leucine Rich Bio Private Limited, which is one of the South Asia's first microbiome company. He is a visionary scientist and entrepreneur with a PhD in life sciences from Indian Institute of Sciences. He is the expert in microbiology, genomics, and microbiome analytics. He is recognized as a thought leader in personalized nutrition and health genomics. He has over 15 years of experience in life sciences and healthcare innovation. He has pioneered in microbiome-based solutions for improving health outcomes. And his achievements are really so many. He's authored several research papers in reputed journals. He's authored several books and chapters. He has several patents to his credit. He has done oral and poster presentations at national and international conferences. He's been the guest and the keynote speaker at conferences on microbiome research in health. And he has been the member of several international scientific communities and advisory boards. Can I have the next slide, please? And he is also featured in many science-based talks on various TV channels such as German TV, ET Now, and Lokmat and Doordarshan from India. And at Leucine Rich Bio, he is revolutionizing the intersection of microbiomics and nutrition. He is empowering healthcare practitioners with cutting-edge microbiome-based solutions. He is collaborating with dietitians to optimize personalized dietary interventions. So it's an honor to have you on this webinar platform with us. Dr. Dhar, a warm welcome to you and over to you.
Thank you. Thank you so much, Dr. Singh, for this nice introduction. This is an honor also for me to be part of this webinar where I will be sharing insights on microbiome with this August audience. I guess and I hope that this session is informative for all of us, and I'll be open to all questions post the presentation. So I would start with presenting the screen. Yeah. So hope the screen is visible to all.
Yes, it is visible.
Great. Thank you. Thank you once again, Dr. Singh. So this webinar is on knowing your microbiome. So microbiome basically is not a very new concept. It's a concept which is quite old. It is quite old in the sense that our old texts in Ayurveda and even Hippocrates, whom we consider as the modern father of modern medicine, father of modern medicine. Um, they all used to say that uh, the health of the human actually belongs, or everything is inside the gut. So microbiome, so what is microbiome? So microbiome basically is the sum total of the genome of all the microorganisms which are present inside and on our body. And in this presentation, I'm going to take you through this universe. I'm going to give you some information, tidbits about these organisms and how they play a role in our health. And I'm going to also discuss a very interesting paper that we published last year on modulation of the microbiome or the microbiota with diet and its implication in health. So microbiome, as I just said, it's now the buzzword in the industry, in the market. But it's also called our second genome, and rightly so, because we are not just the DNA of our cells. We are also DNA of all the other organisms which are present in and on our body. And these organisms play a humongous role in the well-being of ourselves, and hence they are also called as our second genome. So do we consider ourselves as humans? Well, I think the time has come to really smell the coffee, because we are not just humans. We are uh, surrounded by all microorganisms with their genetic material. We also have the microorganisms on our body and inside our body. So if you consider all the genomes which are on our body, inside our body, and in the environment, then the concept of us as being humans is actually it actually becomes quite fake. So we are basically a holobiont, which is a sum total of our cells and the cells of the microorganisms which are present inside and on our body. So with this, there are some of the concepts which ring around us nowadays, mostly, which is the hologenome concept, which I just mentioned, that a holobiont or a hologenome basically considers all the genetic material of all the organisms which are there on the body, which are not in which are basically invisible, but they play a huge role. Then the extended genotype, another organ, second genome, hygiene hypothesis, all these concepts or terminologies revolve around the concept of microbiome. So microbiome has become very important in our days right now. So, how much do people know about the microbiome? You'll be surprised that not many people actually know about the microbiome. And this is a survey which was carried out in 2023 by Biocodex Microbiota Institute and Ipsos, which is a market research organization. It had a sample size of around 6,500 from different countries in different continents. But the general outcome of the survey was that around 21% people knew about the term microbiome, which is fine. It's a technical term. Uh, Interestingly, around 25% knew about the term dysbiosis, which is also fine, because again, dysbiosis is a very technical term. But interestingly, more than half of the surveyed participants had actually tried interventions to decrease the microbiome imbalance. 62% people were aware about probiotics, and I know from my experience, and I think from the experience of this August audience, that any layman who comes to you would know about probiotics. So probiotics is a buzzword which is known to even a 7-year-old child to a 80-year-old person. Another interesting information from this survey was that people in the age group of around 25 to 44 were more informed about gut health as compared to the older adults. And I think one of the reasons why we are seeing this particular section to be much more aware of gut health is because of the advent of social media. So Instagrams and Facebooks and YouTube, they're all full of information on gut health, and hence these people who consume more of these tools in their daily lives, they know much more about the gut health. And finally, 44% people who were informed about the approaches to keep gut healthy by their healthcare professionals. So the healthcare professionals are actually also fundamental in creating the right information metrics for people to understand the importance of the gut health. And finally, uh, and most importantly, 95% people who were informed by their healthcare professionals, they tend to adopt the behaviors to maintain their gut health. So that becomes very, very important, because people like you, people like us, the doctors, the clinical community, all put together have to take this understanding, translate it into something which the general public can use for their well-being. So before we get into the nitty-gritties of the microbiome, um, this is a snapshot of some of the the mega projects that were initiated in different countries. And if you see, this concept of microbiome projects started way back in 2005. Um, you can see here, just hold on. So 2005, which was the Human Metagenome Consortium Japan project. But the project which actually catapulted the whole the journey of the microbiome was this American gut project, which was first of a kind citizen science project launched in various countries, like the US, UK, and Australia simultaneously in 2012. Of course, the Human Microbiome Project, which was started in US in 2007, is also one of the most recognized mega human microbiome projects. And so different European countries and even Japan have been in the forefront of looking into the microbiome. We also know that the Indian government also wanted to start a human microbiome project in India. I'm not too sure whether that project has started yet or not. If you look at the publication trend and the impact areas this microbiome is creating, you would definitely see an upswing. And starting from 2015, this data is still to 2020, you would see that the number of research papers are growing every year. And the kind of areas where the microbiome publications are happening uh, revolve mostly around oral and gastrointestinal diseases, but you would see the cancer is also coming up, and infection and immune system related papers or research articles are also going up. One major improvement or increase rather in the number of publications that we would probably see now to 2030 would be on skin, and of course cancer. So I think these are the areas where most of the research papers are focusing right now, and very soon we would see some interesting solutions coming out from the microbiome domain uh, to cater to the unmet needs of different health conditions that we as humans are facing. So, what is the power of the microbiome? This is a very interesting, slide. If you see, this talks about a particular tribe in Tanzania. They are called the Hasda people. So they are like hunter-gatherer kind of a people, and their gut microbiome is very diverse, and and also very rich, because they have close to 1,200 species of different microorganisms which are absent in let's say a urban dweller. And what is interesting is this particular tribe, they have never ever heard of chronic diseases that we see daily, such as obesity, type 2 diabetes, hypertension, etc. So this goes to show the importance of the microbiome in preserving our health, in keeping us healthy. And hence it becomes very important that we look into this concept in much more depth, in much more clarity, so that we can translate whatever knowledge we have to the general public. So some trivia here. we are generally around 70 kg weight people average. If you look at the bacteria weight combined, it is around zero point 0.2 kgs. There's a misnomer or a false information that has been going on for quite a while now, that the bacterial cells outpopulate the human cells by 10 is to 1 and all those things. Now that has been debunked. It's not 10 is to 1. Yes, they are more in number as compared to the human cells, but the ratio, the proportion, or the ratio is not that high. But what is high is, if you look at the total genome that is contained in the human cell as compared to the bacteria, that is where you see such diversity in the genome. And that's where a lot of possibilities exist in this domain. So we will be focusing this talk mostly on the gut microbiome, although the skin microbiome, the vaginal microbiome, the oral microbiome, they all are very important. But it is the gut microbiome where 95% of the body's microbiota reside. And fortunately or unfortunately, most of the research has been centered around the gut microbiome. So, as I said, the host microbiota and the human body, if you look at it, there are various niche areas where the microbiota reside on the human body. You look at the nasal microbiota, the oral microbiota, the skin, the urinary, the vaginal, and so on and so forth. But we are talking about the gastrointestinal tract microbiota. Here also, if you look at the stomach, the small intestine, and the large intestine, the microbial density differs. So as you go down the alimentary canal, you reach the colon, you see the maximum number of bacteria or the microorganisms residing in that region of our body. This is a quote that I generally present. It says that if eyes are the windows to the soul, then the gut microbiome is the window to your health. And surely, we are trying to look at gut microbiome with much more precision, and it's bringing out some very interesting outcomes, which I'll be discussing in this presentation as well. So, as you know, our gut microbiota is very important, and their importance lie in various aspects of the human health, starting from digestion. So the microbiota residing in our gut help in digestion. They produce something called as short chain fatty acids during digestion of fibers, etc. So these short chain fatty acids, which are basically butyrate, acetate, and propionate, have various roles in our body, positive roles, that is. Then what is very important here is the immunological implications. Many people do not know, but the microorganisms in the gut actually aid in the immune training, immune surveillance, and hence are very important in developing immunity in a lot of in children and in adults also. So that is the immunological implications, and of course their influence on the metabolic processes are well known. They're important in nutrient absorption, in vitamin synthesis, neurotransmitter synthesis, and so on and so forth. So to summarize this slide, basically the microorganisms residing in our gut, especially, they have varied roles. It's not just their presence is just like that. They are there for a reason. And in fact, the evolution that we have seen in humans, the concept nowadays is that it's the holobiont evolution. That means the evolution of the humans along with these organisms which reside in our body. They have co-evolved with us. So you cannot take out the host away from the microorganisms that reside in the host and look at it like that. So it's a combination of factors. So before we get into some more details, let's look at what are the factors that influence the gut microbiome. See, the gut microbiota in general can be divided into various phyla. So phyla of organisms, and this I am talking mostly of the bacteria, because 99% of the organisms in the gut are bacteria, 97 to 99%. Having said that, the fungi, the viruses, the protozoa, everything, the nematodes, all of them play a significant role in the healthy individual. But we are focusing on this slide on the bacteria. And as I said, the bacteria, there are five, generally five important phyla. um, Firmicutes, Bacteroidetes, Actinobacteria, Proteobacteria, and Archaea. And there's one more called Vericom microbiota. So what are the factors that define these the presence of these organisms in various phyla? One is definitely age. So age is a very important factor. Host genetics is a very important factor. So a person X and a person Y will be hosting different microorganisms depending on their age and also on their different genetic profile. So there are certain genes now which are known to help uh, colonize a certain set of bacteria, and the research is ongoing. So the information that we have is limited as of now, but yes, host genetics plays a very important role. The immune system also is a very important role, which define which microorganisms I will have as compared to, let's say, another person will have. uh, Lifestyle and diet, of course, one of the major factors that define our microbiota is our diet and lifestyle. And of course, evolutionary history in terms of gut architecture, how long is the alimentary canal, also plays a very important role, if not a very important role, an important role. And how much antibiotics we have taken during our lifetime also can influence the organisms that reside in our gut. To give you a small example, here on the right hand side, if you see, this is a healthy control. This is just a cartoon. This is not an actual thing, but just to give you a snapshot. And if you see a patient, you have a different, phyla distribution as compared to the healthy in IBD, in type 2 diabetes, and necrotizing enterocolitis, which is a infant, diarrhea kind of disease. So when we talk about the gut microbiome or microbiome in general, a term that comes very often is dysbiosis. So what is dysbiosis? Dysbiosis is basically a disbalance between the organisms that are present in the gut. So we have a very intricate balance of microorganisms, and I'm deliberately using the word microorganism and not just bacteria. However, most of the information that we have in the domain right now is on bacteria, but with active research going on in this field, you won't be surprised to see more information on the fungi and the phages also coming up. But having said that, the dysbiosis that we are talking about is a general dysbiosis where the balance of the good and the bad gets a bit disbalanced, and it causes dysbiosis. So what basically happens here is, again, example is on the bacteria here, that because of dysbiosis, more of the pathobionts abundance go up and the abundance of the symbionts come down, which lead to a lot of clinical manifestations that we see in most of the chronic diseases. So as the shifts the shift towards the pathobionts go up, you see more of the gut permeability, more insulin resistance, and so on and so forth. Whereas if you have the balance maintained, then all those negative aspects of your health can be taken care of, and that's what happens in most of the chronic disease. So this is the impact of dysbiosis. You can not only see in the gut, but because the dysbiosis in the gut, and the gut is linked to various other organ systems. For example, the gut-brain axis is one of the most important axes or axis that we are studying as researchers. So any change in the balance in the gut microbiota can impact your brain and vice versa. Um, and so you have the gut heart axis, the gut lung axis, the gut liver axis, and so on and so forth. You have the gut skin axis. Again, you would see a lot of IBD patients have skin issues. A lot of IBD patients have depression. So that goes to show how a dysbiotic gut can impact an organ which is far from the gut. And that basically tells us that it is not just the gut, it is the whole body that gets impacted when the microbiome or the microbiota are disbalanced. So another aspect of the microbiota that I just want to touch upon here is, can the microbiota be used as potential biomarkers? Uh, 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 it's an active field of research, and people are finding very interesting signatures where this gut microbiota probably can be used as biomarkers in many of the diseases. Some of the snapshots that I have put on this slide basically suggests so. So infant gut microbiome predicts obesity in preteens, which is something which was quite apparent in some of the research. Then gut microbiota biomarkers could help diagnose schizophrenia. Again, schizophrenia and other depression, anxiety, mental health diseases, they do not have an efficient biomarker to differentiate them from other aspects of the other diseases. So I think gut microbiota there can also play a very important role. And various cancers and rheumatoid arthritis, you just name any disease, and people are looking at certain signatures which probably can be used as effective biomarkers in the near future. Which brings me to very important aspect of the gut microbiome story, which is gut microbiome profiling. Now, before I delve deep into this gut microbiome profiling, uh, what has actually made gut microbiota or gut microbiome the buzzword today is the ability to look at all the organisms at one go. Now, see the current, even I must use the word current because still it is the most prevalent kind of methodology in microbiology, is to look at certain microorganisms, grow them in the lab, and then report them. Now there are advantages and a lot of disadvantages with that kind of way of looking at microorganisms. The major disadvantage is that not all organisms can grow in lab conditions. So you will probably able to look at 5, 10, 15, 20 maximum organisms at one go, whereas a new revolution that is coming in this domain is the next generation sequencing, which is the same technology which is used to sequence a human genome. So with that technology, there is no need to culture the microorganisms. Based on the presence of the DNA or the genetic material of the organism, you can predict their presence or their abundance. So that this particular technology has actually catapulted the whole microbiome to the next level. So coming back to gut microbiome profiling, based on the next generation sequencing, there are certain features. One, it is non-invasive. It profiles microbes from the stool. Culture is not needed. It provides relative abundance of microorganisms. Various diversity matrices can be looked into. Can also provide strain level information. Functional detailing can be done based on the microorganisms abundance, presence, or absence. And based on that, what is important here for this audience is that we can provide personalized food recommendations based on the presence or absence of certain microorganisms. Now, gut profiling can also provide hidden clues to existing disease. So, for example, some people might show up as IBD, but many parasites such as uh, strongyloides stercoralis are also they also give symptoms similar to IBD. So, if they can be detected in the gut, then this can be probably rightly diagnosed. So, what is the next generation sequencing all about? Basically, there are two types of technology in use here. One is called 16S rRNA gene sequencing. The other one is called whole genome shotgun metagenomics approach. So, the basic difference between these two is that in the 16S rRNA sequencing, you can profile only the bacteria. Okay, whereas in the shotgun, as the name suggests, whole genome shotgun metagenomics, here you basically can sequence everything which is present in the sample, and hence you can look at the bacteria, the archaea, the fungi, the worms, viruses, etc. So shotgun metagenomics is a much better approach as compared to 16S rRNA profiling. However, shotgun metagenomics is costlier than 16S, and hence in the initial period of the microbiome journey, 99% of the researchers were profiling 16s, and hence more information that we have is on bacteria. But now things are changing. Shotgun metagenomics. Now the researchers are actually doing shotgun metagenomics so that they can profile all the organisms, because it's an ecosystem. The gut microbiota is an ecosystem of different microorganisms and not just bacteria. So it becomes very important that we profile all of them. So to give you a snapshot of how a healthy versus a dysbiotic gut looks like in terms of the phyla level information. If you look at this, this is basically, let's say this is the healthy bar, and this is the relative abundance of the bacteroidetes, which I explained that it's a phylum which contains a lot of different bacteria. And then you have the firmicutes. It's again a phylum which contains various bacteria, especially the lactobacilli species, genus, basically. So if you can just look at this graph, and you will see a differential phyla arrangement as compared to, let's say, healthy and other diseases. Similarly, the another important metric that people look at is the alpha diversity. It's a kind of diversity metric which looks into the richness and or evenness of the microorganisms present in a sample. So if you see, this is the cartoon on or the slide A, basically looks at healthy and all diseased species richness. If you break this into various other diseases and healthy, you would see a variance in different richness profile of various diseases. Um, so this is one interesting difference that one can see when they do shotgun or when they do next generation sequencing profiling. Another aspect of diversity is called Shannon diversity, which looks at the richness and the evenness between the different species present in two different samples, for example. And here also you can see that as compared to healthy, the diversity varies in different disease conditions. So that's the power of next generation sequencing and microbiome profiling. It gives a snapshot of a lot of information on the microorganisms present in the sample. And now we come to very interesting aspect, which is the impact of food on gut microbiota and vice versa. As you all know, food plays a very, very important role in modulating the gut microbiota. In fact, uh, we are born with certain genes which we cannot change. We are born, we acquire certain microorganisms. Fortunately, these microorganisms can be modulated with food, and that's how the whole concept of microbiome modulation through food, diet, probiotics, prebiotics are uh, taking very important interest from researchers and scientists and clinicians and nutritionists and dietitians from across the world. So one very interesting paper that came up in 2022, they looked at the influence of diet, genetics, and gut microbiota on plasma metabolome. So they looked at the different metabolites in the plasma, and they tried to find out which is causing the most variance across individuals, and they found that it is the one of the major variance is coming in the plasma metabolome is through the microbiome. That means microbiome plays such an important role in shaping us as a whole. So a person X is different from person Y. Not only genetics plays a role, but his or her microbiome is also playing a very important role. Some of the interesting metabolites, around 34 of them, were actually coming from the microbiome that he or she possesses. And of course, through diet, the majority of the metabolites were present. As we all know here in this audience, I don't have to elaborate on this. Vegetables, fruits, whole grains, plant-based food, certain fishes, uh, and certain fermented products, they all are considered to be good for our gut microbiota, mainly because of various reasons. They basically help the good ones to proliferate and the bad ones to go down. Whereas when you look at the high fat dairy products or processed meat, or rather processed food, they all lead to conditions which is very dysbiotic in nature, and can cause lot of harm in short to long term consumption. So this is something that is well known, but the fact that these food have an impact on us based on the role they play on the microbiota is something that we have to keep in mind. Again, this is a very interesting paper that came out in 2023. So they looked at the different aspects which can impact the microbiota and this different chronic health conditions. Here if you can see around 101 papers that they looked into. This was a meta-analysis. They found that diet played a very important role in obesity, then metabolic syndrome, some aspect of type 2 diabetes. Probiotics, interestingly, also played quite a good role in obesity conditions. Prebiotic also played a very important role in obesity conditions. So basically, it's a meta-analysis which showed that all these different modulators, uh, and different, chronic diseases, what is the link between them? And this is how they found that the link of diet mostly on these chronic diseases. When they looked at these parameters and how they impact the gut microbiota, again, very interestingly, diet was found to be the most important factor that had an effect, a positive effect on the cardiometabolic traits, as well as an effect on the gut microbiota. Interestingly, probiotics, which we all know about, they had a very minimal effect as compared to the diet and the prebiotic and symbiotic, etc., on the impact on the gut microbiota, whereas they had a pretty good impact on the cardiometabolic traits that these researchers were looking into. Again, another interaction which came up in Nature Medicine in 2022. Here basically what it shows is the impact of the diet on gut microbiota and vice versa. So on the left hand side of the slide, you can see that increased coffee intake increases hippuric acid, and hippuric acid is known to modulate Methanobrevibacter smithii, which is a bacteria of a lot of importance. So this is an example of how an ingredient of a food can impact the gut microbiota. On the right side of the same slide, you would see how a particular microbiome species would impact a certain metabolite. So here, when you take a lot of fruits, and if you have presence of certain organisms, especially ruminococcus, then a certain metabolite called urolithin B is modulated. So this is how the gut microbiota impact food, and food impacts gut microbiota at the molecular level. Which brings me to the next part of this presentation, which is on this particular paper that we had published last year. So this was the first paper of its kind coming out from this part of the world, where we showed that if you modulate the microbiota based on personalized nutrition, then it can improve hyperglycemia and hypertension parameters. It was a prospective open label randomized clinical trial, and I will take you through the outcome of this particular trial, and try to explain the rationale behind the trial and what we found and what we achieved. So to give you a brief of what was this trial all about, so we basically took 30 patients, 30 type 2 diabetic patients. They were divided into two cohorts, test and control. In the control, these patients were given a dietitian approved meal plan, and they were asked to continue with that meal plan for 3 months. And we after 3 months we looked at certain parameters like HbA1c, blood pressures, inflammation markers, and so on and so forth. In the test cohort, we basically gave them a particular gut microbiota uh, based nutritional meal plan, which they were asked to continue for 3 months. Please understand that in this cohort, all these patients were given their personalized nutritional meal plan based on their personalized gut microbiota profile. So every individual got, although all of them were diabetic, but all of them got a different kind of a meal plan based on their gut microbiota profile. So that's the novelty of this particular trial that we tried to do. And after 3 months we again looked at certain parameters. We also looked at their microbiome profile pre and post the intervention. So very interestingly, what we found was that the HbA1c level, which is a marker for the glucose in the body, it had come down in the microbiota modulated nutritional cohort as compared to the regular nutrition arm, and this was statistically significant. There was a certain decrease in HbA1c in the control arm also, but this might be because both the arms people the patients were given stabilized dose of anti-diabetic medicines, because we cannot remove the medication for these people, and they were also given certain and they were also following certain dietitian monitored meal plans. However, the difference here, or the shift in HbA1c, was not statistically significant, whereas the shift here was statistically significant, and the percentage decrease was also higher. So if you can see here in this next slide, I have put the microbiota modulated nutritional meal plan cohort. uh, we had around 20% decrease in the HbA1c level, whereas in the regular there was around 10%, and that also was not statistically significant. When we looked at the individual decrease of HbA1c in this cohort, in the test cohort, we found that all the individuals showed a decrease in HbA1c, whereas in the control there was decrease in certain individuals, but there was increase in certain other individuals also. So that means it was not very uniform, and that's why it was not significant also, the decrease. Next, we wanted to look at the impact of the microbiota modulated nutritional meal plan on hypertension parameters. So when we looked at it, we found that again in the regular cohort there was slight increase in the systolic pressure, average systolic pressure, whereas there was 5% decrease in the microbiota modulated cohort. Then we took a subset of the individuals who had a higher hypertension parameter at the baseline. The baseline that we selected was more than 130 millimeter of mercury. When we looked at that cohort, we found that there's a 14% decrease in systolic pressure in those patients, which was statistically significant also, and diastole also there was around 4.5% decline. We also looked at the inflammation markers, and CRP was one of the markers that we looked at, and we found around 20% decrease in pre as compared to post in the CRP level, suggesting that even inflammation went down in many of these patients who were in the microbiota modulated arm. Then we looked at the microbiota changes at various levels, at the phyla level, at the genus level, at the individual species level also. What we found that in individuals who had taken in the test arm, there were certain changes in the phyla also. If you see here, in the post arm, the same individuals, we found that there was increase in firmicutes. If you remember, I told you firmicutes is that phyla which consists of various bacterial genuses like lactobacilli. Many of our probiotics actually come from this particular phyla. So that is an interesting thing that we observed. We also saw an increase in actinobacteria. Again, actinobacteria is that phyla which contains bifidobacterium genus, and as you all know, bifidobacterium also is one of the very important probiotics. And interestingly, there was a decrease in bacteroidetes. In the pre, you see this is the green is the bacteroidetes, which has come down here. Again, bacteroidetes contains various bacteria, but mostly they bacteria like Prevotella, etc. They belong to this particular phylum. So when we looked at the genus level, we found again in the post there was increased Rosoburia. As you know, Rosoburia is a very interesting genus. It has been linked to positive health benefits. We found a decreased Prevotella. As you know, Prevotella again has been found to be associated with inflammation. So we found that Prevotella has come down in the post phylum analysis, and in decreased Ficacola and so on and so forth. So then we looked at some of the individual species which we found them to be differentially abundant, either positively or negatively. Now, interestingly, we found certain species of bacteria which were reduced in abundance. We found Suterella, we found Alistips. Now, Alistips Feingoldii before has also been implicated in high blood pressure. And if you remember from the presentation here, that we also found that the systolic and diastolic pressure had actually come down in a lot of our subjects in this cohort. So, which correlates well with decreased Alistips Feingoldii. And we found some interesting increase also of certain abundance of certain microorganisms. Most importantly here is Bifidobacterium angulatum and Levibacillus brevis. So basically these are also known probiotics, and this was the first report where we showed that Bifidobacterium angulatum is actually going up after the intervention, which is microbiota modulated intervention. Then we looked at some of the alpha diversity matrices. Remember in the earlier slides where I was showing the power of the microbiome profiling. So basically you look at certain diversity matrices. When we looked at the diversity matrices, we found that the diversity of the bacteria improved in our test cohort post as compared to pre, whereas other kingdoms we did not see much difference in their kind of diversity. Overall diversity, when we looked at, we found that the overall diversity, the Shannon diversity actually had increased in the post cohort as compared to pre, which was very interesting. Whereas the overall richness was slightly lower in the post cohort as compared to pre. Now, this might seem contradictory, but it is not. When we looked at Shannon, see, Shannon, as I mentioned, is a parameter which looks at richness and evenness of the microorganisms, whereas this Chow one basically looks at only richness. So that means that with the intervention, the diet intervention based on the microbiota of the subjects, the evenness of the microorganisms has improved, and the richness has slightly decreased. And it's the same kind of pattern that was found by another group before, where they looked at multiple disease areas and they wanted to look at how the diversity matrices varies between healthy and disease groups. They also found similar things where they found that the diversity overall had improved Shannon, and the richness had decreased a bit in the healthy as compared to the diseased. Then we looked at the network analysis. We looked at how different microorganisms are interacting among themselves in both the pre-intervention group and the in the pre-intervention as compared to the post-intervention. Now, here in this network analysis, if you see, the red bars are the negative correlation, and the green bars are the positive correlations. Now, I want to draw your attention to this particular section of this network analysis. Here, you would see that there's a bacteria called P. macillensis. This one, this is negatively correlated with another bacteria called Suterella. Now, alongside, P. macillensis is is in positive correlation with another bacteria called Parabacteroidetes distanesis. Now, Parabacteroidetes distanesis is a very is called the next generation probiotic by many. It's a very I mean, it has positive outcomes in the health of individuals. But we found that in the pre-intervention, uh, Suterella is negatively correlated with this guy, and this is positively correlated with this guy. But in the post-intervention, that Suterella, that negative correlation had actually disappeared. And if you remember, if you see from our differential abundance also, you would see that Suterella in the post is completely gone. Okay, so this is a speculation. We had speculated in the paper that it is possible that because Suterella was under a negative correlation with Macillinus, higher Suterella was leading to lower Macillinus, and lower Macillinus therefore was not able to improve the abundance of dystonesis, which is one of the important bacteria for health. Whereas, so that is what is being highlighted here in the post-intervention slide. There's another thing that we wanted to highlight is that the role of succinate. So people have shown that succinate is important in modulating hyperglycemia, but higher succinate level leads to inflammation. So a theory that we proposed in this particular paper or in this particular trial or study was that it is important in conditions like type 2 diabetes that the succinate metabolism should be balanced. And we found that there are certain organisms, for example, Acinetobacter, which this one, P. succinatosus. Even I cannot spell these organisms, but P. succinatosus have been known to consume succinate and produce propionate. So the theory that we proposed in this particular trial or study was that one of the reasons why we saw an improved hyperglycemic condition in patients who had taken the intervention was there was a better metabolism or balance of succinate. So the succinate consumers and succinate producers were in balance, whereas that was not the case probably in the other cohort. Okay, so another very interesting, again, this is just this is a cluster analysis where we did a hierarchical cluster analysis. If you look at the pre and compare it with pro, you will see that there's a lot of clustering happening of the microorganisms, whereas that cluster is not visible in the pre before intervention, that is. We do not know the reason why this is happening, but this is an interesting observation, and we thought we will we must put it in the research paper as well. So this is how we summarize the trial. We saw that in the trial, people with type 2 diabetic, the type 2 diabetic patients with high systolic pressure and high inflammation, when they were given microbiota based nutritional meal plan, they had an improvement in their HbA1c, in their hypertensive parameters and inflammation markers. The various mechanisms at play can be balanced succinate metabolism. That is something that I was explaining in the previous slide, and also abundance of certain probiotics like Bifidobacterium angulatum and Lactobacillus brevis type of organisms. So all put together, you see the effect that we see in this particular study. So now I come back to the overall aspect of the microbiome. So microbiome can have a major impact on healthcare. It can improve diagnosis, it can improve prognosis, and it can improve therapeutics. So with improvement in all the three parameters as you see above, we are basically going from sick care to wellness. And since we are talking about various interventions, I thought let me share some important interventions that are available right now. We have heard about FMT, which is fecal microbiota transplantation. So the advantage is that it is effective in modulating the microbiota because you are giving the complete ecosystem from the donor to the recipient. But it's highly complex, and it prevents standardization because every donor is different, every recipient is different, so the standardization becomes difficult. Then you have the FFT, which is a step or another version of FMT, where you use the filtrate and not the complete sample. So it is reduced chance of infection because you are using a filtrate, but again it is very sensitive to preparation protocol, and it is still under research. Then you have the diet, and one of the examples that I just shared of how diet can play a very important role. Personalized diet, not diet based on certain conditions, but diet based on the microbiota. So people have shown various aspects improvement in various conditions like the paper that we just discussed, but it is difficult to change habitual eating habits, and difficult to follow the meal plans and so on and so forth. Then you have the fermented foods, you have the prebiotics, you have the probiotics, then you have the phage therapy. There are certain companies which are working on the phage therapy aspects also. But again, the problem there is that the lab grown phages might not adhere to their intended targets in the actual condition. So it's something, again, the research is happening, and hopefully we should see better products in that category. And of course you have the postbiotics. Basically it includes all the secretions as well as the part of the probiotic which is making the postbiotics. So if you look at it, basically all these interventions look at either species introduction, species depletion, or enhanced growth of certain organisms. Now, since we talked about therapeutics, I'll just give you a snapshot of what is happening in the therapeutics arena. So there are few companies, microbiome companies like 4D Pharma, Finch Therapeutics, Vedanta, and Series, and they have collaborated with some of the bigger pharma companies, because bigger pharma companies also want some new novel solutions for many of these diseases. And as you can see from this particular slide, a lot of these conditions, microbiota solutions are being looked into. And finally, this slide basically gives you the good news that now there are two products that have been approved by the FDA, and both of them basically are used to treat recurring clostridial difficile infection. So these are the two products. One is the Rebiota's or Ferring Pharma's product. The other one is called Voust. It is the Ceres Therapeutics product. The difference between these two are shown here. Basically, Ceres Therapeutics products Voust is a oral capsule, whereas the Rebiota is basically a liquid enema based product. And there are other companies like Vedanta which are working on different aspects as we saw in the last slide. So at least two products are in the market in the therapeutics domain, and hopefully in the next half a decade we will see many more products coming out of the microbiome therapeutics domain, and addressing many different diseases and not just Clostridium difficile infection. With that, I come to the end of the presentation. I thank you all for your patient hearing, and I am open to all the questions now. Thank you so much.
Thank you so much, Dr. Dhar, for an insightful session. While I was listening to you, there are so many questions coming into my mind to ask you and understand how you do this. So there are a lot of questions on the chat box also. I would request Ms. Shalini to come over and start with the Q&A session.
Sure.
Thank you, Sandhya. Thank you, Dr. Da. It was very, very insightful session. And the session shows, your slide shows how very passionate you are with your work and many logistic which has gone behind. So there are few new things which were there for us to take, like next gene sequencing. As you said, not all microbes can be grown in the lab. So this is something new, which even without culturing, we can get to know many things,
Yeah.
and also a relief about C. diff bacteria, which is quite can be very fatal and notorious. And it's good to know that there's some pill coming for that. And a few questions from our side. First thing
Sure.
is, as we know that any microbiota for every person is like a fingerprint for a person, right?
Yeah.
So
Yes.
how advantageous it is for a person who has not been on, say, on antibiotics. So after antibiotics, probably there'll be a dysbiosis for a while, and doing intervention would be fine. But for a normal person, because there are so many products now coming with different, products with different genes, culture. So is it good for people to take prophylactically, say for 3 months, taking probiotics and things like that? What's your take on that?
So yeah, see, taking probiotics in general, it can do two things. Either it will not do anything, or it will be of some positive impact. So there's no harm in taking probiotics as such. But if you're healthy, um, then there's no need, because most likely your gut is fine. So there's no need unnecessarily to to take certain probiotics.
Okay.
But the again the conundrum is, or the problem is, you do not know whether your gut is healthy. Just because you do not see any clinical manifestation does
Hmm.
not mean that your gut is healthy. And in fact, we have seen this. We have seen in many conditions where the gut, there's huge dysbiosis, but that particular person is apparently healthy because the clinical manifestation has not yet happened. So yeah, prophylactically you can take probiotics. It's not going to harm you, but don't take probiotics for too long, because again, taking anything in excess is not good.
True.
So maximum what it can do is not do anything, if you take for 1 month to 3 months. But beyond that, I would not suggest unnecessarily taking it.
So, yeah,
um, yeah, do a profiling if required, just to see how things are.
correct, correct. So it doesn't take over the already existing microbiota, right? If we take
See,
probiotics like that.
no, so it's not a very easy answer actually. It's a very good question, and there's no easy answer to this, because we don't know. We don't know. See, it depends on so many factors. Number one, it depends on the which strain of probiotics you are taking, because not all strains are same. Second, it depends on the colony forming units, or the CFU counts. Now, if you are taking probiotics which are of low CFU counts, the chances of them colonizing your gut is very, very minimal. Third is that how these probiotics are being delivered. If they are delivered like in sachets and all, where the chances of the probiotics getting destroyed in the stomach is very high, then again the
Hmm.
colonization will be very, very limited. Fourth,
Hmm.
we do not know the genetics. Again, that's something that research is trying to find out. But there's a possibility that a person harbors a particular gene, and I'm talking of an extreme case actually, which probably will not allow a certain probiotic to colonize. Okay, so we do not know that aspect too
Okay.
much right now. So
Okay.
there are so many
Okay.
variables right now to give you a straight answer to this. But yes, my I always say that there are two things when someone is taking probiotics. One, it should not be given to a immunocompromised person, because we have seen that in many cases that a person who is immunocompromised, unless a particular probiotic is necessary for that particular
Hmm.
individual,
Hmm. Hmm. Hmm.
generally it should not be given. And
Okay.
and secondly, yeah, the duration of the probiotic should be monitored well. It is more of a trial and error basis. You might be you might take certain probiotics for some time and see the effect. If it's giving you a good
Right.
effect, you continue for some time and stop it. More importantly,
Right.
take prebiotics for the probiotics to grow.
True. Correct. Correct. And how common is FMT in India? I
See,
mean,
FMT
we
is
see
not
very few cases. Yeah,
very,
but
yeah, yeah. So FMT is not a regulated kind of a practice. People are doing FMT under the radar, I guess.
mhm.
We see very limited FMT studies on clinical trial websites. But that
Mhm.
that does not mean that people are not doing FMT if they are not putting it on the website as a trial study. I know many places or some places where people are doing FMT quite regularly, but again, it is something that needs to be done in a very proper manner. I'm not sure how things are
Mm,
right now in India.
true. And in your practice, how many physicians, I mean, it might be a little vague, but have you come across many physicians who are also prescribing probiotics after antibiotics? Because this, I think it's yet to be caught up by physicians where after
Yeah.
antibiotics, giving probiotics. Have you seen that in practice?
Yeah, I have seen a lot of people, lot of physicians giving that. So there was an era when people were giving only lactobacilli, as probiotic uh, after antibiotic or during antibiotic. Now people are giving more combination of probiotics, because again, giving one strain, one species might not suffice. So yeah, I'm seeing that trend of physicians giving probiotics more uh, after an antibiotic
Okay.
course.
Okay. Let me take a few questions from the chat box. Okay, so Dr. Sandhya is asking, I have a question. What is the sample that you would would harvest to understand individual microbiome?
I didn't understand the question, frankly. What is the sample? I didn't understand.
What is the sample that you would harvest to understand individual microbiome? Dr. Sandhya, can you please clarify your question? Okay, while she comes back, uh, okay. How would you measure the interaction of the microbiomes in the individual's body? This
Yeah,
is another question from Dr.
so,
Yeah.
so interaction is basically through bioinformatics. So, so there are certain algorithms tools that are used. There are certain machine learning tools that are used. Correlation analysis are used. So basically, so those tools are used to look at the interactions between the microorganisms.
Okay. And another question from her. How much time is needed to study the impact of the personalized diet to enhance microbiome post-intervention? And for how long will it last?
So very good question. Again, there's no straight answer to this question,
Hmm.
because so every individual is different. As you started by saying that microbiota is like a fingerprint, right? It,
Okay.
it is so different. So in some individuals, the impact of the intervention starts to be seen within, let's say, 15 days, whereas in some people it might take 3 to 4 months. So on an average, I would say around 3 months should be given for the intervention to show its effect, because ultimately what you are doing is trying to change the ecosystem to some extent.
Hmm.
And once the ecosystem gets changed in a sustained manner, then only probably you will start seeing the effect, because they will start secreting certain metabolites which will have an impact on certain parameters. So I think, of course, there's no rule of thumb here, but 3 months to 4 to 5 months, I would say, is required for effective remedies or
Changes.
solutions. Yeah.
Correct. Correct. Okay. In your career, have you seen any improved quality of life and better tolerance to food in patients with IBS and IBD?
Yes, I mean, we have a lot of patients who take our test, and some of them have reported improvements. But the problem with us is that we don't follow up with patients, and we don't keep following, because they do the test and they go away. uh, Very few would come back and say that things have improved or not improved, because they don't keep us in the loop. But
Okay.
there are certain, and that was one of the reasons that we wanted to do the clinical trial, because on
Okay.
type 2 diabetes also we were getting a lot of anecdotal evidences. People used to come back and they say, I know, I'm feeling better, my HB1C is improved, but we wanted to
Hmm.
prove it in a clinical trial setting, and hence we did that.
Yeah. And also, just out of curiosity, have you seen any study where we have done the comparison of microbiota of our generation as to our elders? Because there's so much of adulteration in the food we eat.
Yeah,
Obviously,
it
density is
has been
lesser.
done. It has been done. These kind of studies
Uh,
are done worldwide, actually. Not only that, people have shown that centenarians, or people who live more than 100 years,
mhm.
their microbiota is like a young individual. So their microbiota is does not behave like an old person's
Hmm.
microbiota. So these kind of studies have been done
So
and are
clear.
being done.
Okay. Okay. Um, I should be able. Any more questions? Yeah. Who do you think the physicians are not caught up with the gut microbiome test? No, why do you think that the physicians are not caught up with gut microbiome tests?
Yeah, so there's the many reasons. One of the important reasons is that this concept is new. This concept, in the
Hmm.
sense, the next generation sequencing. So I'm sure in the MBBS curriculum, gut microbiome, role of gut microbiota is not well defined right now.
True.
This will
True.
change as the field advances. Secondly, I think
True.
lack of clinical trials, effective trials to show the impact of modulating the gut microbiota in various diseases is missing. So
Hmm.
so slowly, as we have proved in type 2 diabetes with our kind of thing, more such studies will be done, and then I think the physician community will take a note of it, and then they'll start putting up alongside their medication, so more of a supplementary rather than anything else. So yeah, so
Yes.
that's basically the answer.
Which which sample will give us an accurate representation of the microbiome in our colon, for example, stool or blood?
I think it's tool, because you are
Stool.
looking at,
Mhm.
yeah, yeah.
Okay. Are there chances that the gut microbiome profiling can become less expensive?
Absolutely, absolutely. So, so there's something called as Moore's law, where
Mhm.
it was suggested that the rate at which the next generation sequencing cost is going to come down. So I'll give you an example. When the Human Genome Project was started or initiated, it took 13 years and $1 billion to sequence a human genome. Correct. Now the same can be done. A human genome can be sequenced in less than 24 hours and around
Mhm.
$1,500. So that's the rate
Mhm.
at which this technology is progressing and the
Mhm.
cost is coming down. I'm very sure within the next 1 year at least, we will see a drop in the price of the microbiome sequencing by half.
Are there any more questions coming in? Okay. Are gut-based therapies common in to treat neurological diseases?
It's not common yet, but it is coming up. So because, see, neurobiological disease or neurological diseases have lot of unmet medical needs, starting from diagnosis, prognosis to therapeutics. So gut based therapies or gut based solutions are being looked into, and I'm sure they're going to be much more common as we move ahead.
Then question from Vidya Pujari. Any findings regarding the impact of sports supplements on gut microbiome?
Yeah, I mean, we have not done it, but there are studies where people have done those kind of studies. In fact, not on one of the studies that I'm aware of, where they had actually looked into the gut microbiota of high profile athletes and compared it with the gut microbiota of, let's say, healthy normal people, and they found that the gut microbiota of high profile athletes are absolutely different
Mhm.
from healthy individuals. So these kind of studies are being done quite regularly across
Right,
the world.
right, right. Can food allergies be treated or managed by gut microbiome intervention?
So this is something, I think the science is still very nascent. So people have used probiotics in treating certain allergies, and they have found some positive results also. But I think the science is still nascent. Can it be done? Yes, it can be done. Will it be done? Probably it will take some time when we understand more of how the interactions are happening, and improvements can be done.
Okay. From Sheena Nambiar, is it okay to take different probiotics and prebiotics and probiotic tablets at the same time, or should there be a specific time gap between taking them?
See, again, no rule of thumb as such, but see, what are prebiotics and what are probiotics? So prebiotics basically are food for your probiotics. So I don't know. I mean, I would not be able to give you a straight answer on this. Um, it will probably depend on the individual also, because some, see, one thing I can tell you, not all prebiotics will give you same impact. So for example, if I'm taking inulin or arabinoxylan, for example, or FOS or GOS, anyone, um, I might be impacted differently as compared to, let's say, another person. So that means what? That means that for the prebiotic to show an effect, there has to be some basal level of microorganisms present in the body, and that's why you see the variance. So again, very difficult. See, in microbiome, it's very difficult to give you a straight one size fits all kind of an
True.
answer. So I would not know. Is it okay? Maybe, maybe not. I don't know.
Yeah. It's like nutrition. Many facets
Yeah,
of
yeah.
answers will be there.
There are so
Yeah.
variable,
And
the variables are so many,
true.
so you don't know.
True, true, true. And also, I had come across something where after a person has undergone FMT, due to some reason, either IBS or I think she had CA colon, I don't remember. But then the person who was doing the podcast, he said that the behavior, also eating behavior changed. Earlier, he was underweight, he hardly had any appetite. And after the fecal transfer, his appetite changed and he actually became an obese person.
Hmm.
So
Absolutely.
that's quite possible. Okay.
I have come across many such cases for FMT.
Mm-hmm.
One interesting thing,
Yeah.
since you raised FMT thing, is that FMT can be effective in many diseases or many conditions, but what is very important is what you eat post that. There was one case where a person had IBD, and the IBD got. There was remission of IBD after one or two sittings of FMT, but then that person started eating out a lot. He started eating a lot of
Mm-hmm.
Western diet, and IBD returned. Okay, so
Mm-hmm.
see what is happening in FMT. You are, as I said, you are taking the whole consortia of the ecosystem from the donut and just input in the recipient. But for that consortium to grow and sustain, you need proper food.
True.
So if you start giving them that food, it's okay. If you start giving them, starting antibiotics, for example, or start
Correct.
eating
Correct.
the food, then those get destroyed again.
Obviously. Correct. True. Very true. Okay. What are the different tests we can do in order to know about gut microbiomes?
So one is the gut profiling, which I can suggest. See, there are other tests like calprotectin test is done, which is basically to look at the inflammation in the gut. Then there is lactulose mannitol test that is done to look at the gut permeability. Leaky gut, as some people would call it. So those kind of tests are done generally. But the microbiome profiling is a much more in-depth kind of a test or profiling. So that is something which is better, I guess.
Okay. Okay. This is next question from Sheena. Is a microbiome test useful in IBS? Yes, it's I think you have
Yeah, it
already
should be.
answered.
It should be, yeah.
Yeah.
Because see, what happens is in most IBS and IBD patients, especially in IBS patients, because it's not defined properly, there's no biomarker as such. So when once you hit a dead end while treating these, diseases, it's very important to know what is happening in the gut. What are the microorganisms which are residing, and why are they residing? Is there a way we can improve that balance? So I think, yeah, of course.
Okay. Can Yakult be taken once a week regularly?
I don't know. Maybe I don't know.
Yeah.
So
Uh,
Yakult basically has one strain of bacteria, Shirota,
mm,
and see, Yakult is a very old company and thing, but now uh, things have evolved.
Living.
People have combination of probiotics have also come up, so no harm. But I don't know. I mean, I personally do not
Yeah.
know.
Yeah. Okay. Please share the diet suggested for different microbiomes based on your study.
No, no, no. So this was basically based on the personalized microbiota profile of each individual. So there were 15 individuals. Each of them were given a diet based on their microbiota profile. So there's no common microbiome based diet that I can
Diet.
share.
Could you please suggest any corpus indexed journals which are favorable for publishing research based on gut microbiome, probably which are good journals, easier to publish with? Vidya Pujari.
Easier to publish, I don't know, because publication is a different ball game altogether. Your study has to be good, and everything has to be proper. There are many journals, like you have the BMJ journal. You have there's a journal called Gut. That's also very good. We published in PeerJ, so PeerJ is also very good. There are a lot of Elsevier journals which are good. Nature Springer publications are there. So there are many. There are many actually. So
Okay.
you have to basically see the context of your study, and which journal suits that, and based on, and then, of course there are impact factors and other criteria. Higher the impact factor,
Correct.
the higher the chance of rejection, and all those things are there. So yeah.
Okay. How much time is needed to study the impact of the personalized diet to enhance
This is
microbiome and
done.
intervention, and how long? It's already done.
This was done.
So, how many more questions? Yeah, I think with the interest of time, can we take two more questions?
We'll take two more and we'll be done, shall we?
Yeah, yeah, yeah. So, probiotics effect on hormones defect, specifically which lead causative factors of obesity, which lead causative. Okay. So,
um,
what are
I
the
could
different effect?
understand the question, but I can tell you that yes, there's an impact. So probiotics have been tried for obesity also, as you would see in one of the slides I had shown, and various hormonal defect uh, conditions also. So probiotics have been tried for many conditions. So I don't know what it means, this particular
Probably
sentence.
the effect of probiotics on hormones is what the person is meaning.
Yeah, that's what. So hormonal deficiency or
Yeah.
hormonal defects, probiotics have been
Mm-hmm.
tried,
Right.
and there have been mixed results.
Right. Okay. I think that's all. We have no more questions. Sandhya, you have any?
Yeah. I have one question, Dr. Dhar. There
Sure.
are, different kind of probiotic products available in the market.
Yes.
One is for gut, and one is for women, and one is for children. Something like this they are marketing right now. So, is it needed that it should be, given like this for specific conditions?
Okay. Again, no straight answer, Sandhya, but so there are certain strains where people would have done a trial and have shown that those strains are important, let's say, for bone health, right?
Okay.
So or let's say in improving your hyperglycemic parameters, right? For example, there's a bifidobacterium breve species, a strain, a particular strain which people have shown that it decreases hyperglycemia in certain people. So then certain companies market that for that particular trait or condition. um,
Understood.
Other than that, I don't see any specific reason why they would do that.
Okay, I understood the point. Thank you.
Yeah,
Thank you so much.
Right. Thank you, Dr. Dhar. I think you took so much time in explaining. Your slides were comprehensive, extensive, and you took so much time patiently asking and answering our questions. Thank you so much. We all are
thank
one
you.
step bit more towards knowledge in this new field. Thanks a lot for sharing your thoughts. Thanks to Dr. Sandhya for taking all the hardship running around for all these sessions. And thank you, Aisha, for the courtship and arrangements of streamlining. Thank you so much. We look forward to learn again from you in this topic with few more insights in the future. Thank you so
Sure,
much,
it was a
Dr.
pleasure. It was really my pleasure, and thank you to Dr. Singh for inviting me for this particular webinar. And I hope to be part of this again in the future. Thank you so much.
It was a pleasure. Thank you
Thanks
so
to the
much.
audience.
Thank you.
Thanks to the audience too. Thank you.